Next up, we will have Dr. Scott Bodridge from West Virginia. Thank you for joining us, and he will be discussing St. Croix sheep as a model for immune function and parasite resistance.
Please feel free to get up and leave at any point if the immunology gets too deep or it just becomes alphabet soup. I totally understand that. It's been a long day with all of that stuff. So thank you for having me come out here to Utah to talk about St. Croix sheep. When I first started, so I grew up in Southern California out in the desert, San Bernardino County. Grew up raising Rambouillet sheep out there, showing sheep all over the Western US. Was a high school ag teacher in Central California for a little while before I moved to Maine and started getting into parasite issues in sheep. And I was working for a nonprofit rare breeds farm that was founded by the guy who invented the Ethernet. So the little plug in the back of your computer that connects everybody. So we had a fair amount of capital to work with and managing livestock in Mid-Coast Maine. And I ran into some folks that were doing some work there with Katahdin sheep, and that got me started in that part of it. Before I went to Virginia Tech and worked with Dave Notter there and Dr. Zajac and a bunch of those folks that were doing this. So I want to start this by just kind of going back a little bit. Oh, there we go. And just talking about this parasite problem, right? So as a kid growing up in the desert of Southern California, parasites were not a big deal for us. And I do remember my father very specifically when ivermectin came out, he was so excited to be able to deworm pregnant ewes. I don't know what he was excited for. Nowadays, I just think about, why was he deworming anybody? I mean, we didn't have irrigated pasture. We didn't have much pasture at all. We bought a lot of hay, right? And so when I was in Maine, I was giving some folks who were giving a talk like this some static about that. And so goes the story. So the life cycle of these worms it's very direct and it's very simple, right? The host and the sheep in this case consumes larvae that are on the grass. They develop into adults within 14 to 21 days after being inside the animals. Then they start producing eggs. So the adults mate, they produce eggs. That female is going to produce anywhere between 5,000 and 10,000 eggs per day. Those drop out onto the pasture, and in the correct environmental conditions, which the island of St. Croix is the correct environmental condition for Haemonchus, where it's warm, moist, and humid pretty much all year round, those larvae go through this development stage, which takes about 7 days in the environment before they can be consumed again by the host. So thinking about all of this and putting these things together, what does Haemonchus do to the host? Just kind of give you a sense of what goes on here. Once they go into the host, they're going to raise the pH of the abomasum. So the abomasum, or the fourth compartment of the ruminant system, is where chemical digestion occurs. A high acid environment. So it's going to raise the pH, making it more basic. It's going to suppress immunity. So those worms produce little molecules that suppress the host immune system so they can survive. They're mobile. They have no hooks around their face or their head, so they can move to different spots in the abomasum and continue to feed. And every worm is going to take about 50 microliters of blood per day. Not a whole lot. It's about the amount of water that you could hold on your fingernail. It's about 50 microliters. Multiply that by 1,000 worms. Now you got 50 milliliters. Small lamb, deadly, 200-pound ewe, not terribly costly, but that's what these guys look like. If you've never seen Haemonchus in its native environment, you can see some video of that worm moving there. So everything's moving around there. You can see why they're called the barber pole worm as it gets closer, and we don't have as much sunlight coming in, but you can see that twisted pattern of their stomach around their ovary on the adults. So because they're feeding on blood, their ovary is white, their stomach is red. That's why they're called the barber pole worm. If you never understood why it was going on, there's your debunking or just some explanation of what's happening. All right, so why is this a problem and why does it matter? This is old data. This is from 2008 out of the Ray Kaplan's lab down in Georgia, where they looked at resistance status on different farms to benzimidazole. So those would be white dewormers like fenbendazole, Panacur, Safeguard, Valbazin. those type of drugs, levamisole, ivermectin, moxidectin, so that would be sidectin. So in 2008, there was a lot of susceptible farms, and these are sheep. Well, we can just look at sheep farms for this case. 19 out of the total number of farms there were still susceptible to moxidectin. Moxidectin and ivermectin are in the same drug class and have the same mode of action. So they kill worms the same way. They're just, moxidectin's a little bit more potent than ivermectin is. Benzimidazole is complete resistance, right? So 43 out of those farms, it just doesn't work, right? So this is why parasitism matters. Parasitism matters because I got a call from a producer in South Dakota, a fifth generation rancher in South Dakota that lost 200 lambs in 1 month. Put them out on pasture, gave them a benzimidazole, so a white dewormer, and lost 200 lambs in a month. That's why it matters. Is it just in the Southeast? No, it's all over the country. We did work in Central California with one of my friends on my judging team in college. She needed a high school science fair project for her kids at the school she was teaching at. We just decided to do this on club lambs that are grazing irrigated pastures in Central California. The fecal egg counts went up after they got benzimidazole treatment, went up by 40%. They only went down by 16% with ivermectin or sidectin treatment. So it's a serious issue. And it's not just in my neck of the woods, it's all over the country. So when I got to Maine and we're studying this stuff, I was looking at Katahdin sheep at the time. And, you know, everything that you read about all of this stuff, everything has to do with the animal clearing the infection. So anytime that we're talking about clearing an infection, we're talking about immune response in those animals and how they're able to respond to this. So Courtney's study, thank you, Dr. Wildius, for bringing all of that stuff in there. Dr. Courtney down at the University of Florida and Dr. Parker at Ohio State University in 1985 showed this big difference of St. Croix sheep and Barbados blackbelly being really parasite resistant. That became one of those fundamental papers that everybody started looking at. Then a few years later, Dr. Zajac, while she was doing her PhD during her DVM at Ohio State University, did a comparison of St. Croix and Dorset sheep. Just thinking about Dorsets being a typical Midwestern type sheep that was going on in Ohio at the time. And what they found is that the St. Croix had lower fecal egg counts than the Dorsets, right? Now we can say that. At the time, they weren't certain that that was going to happen. But what they also found was that there wasn't this big immune response difference that they expected because it was so far past when those animals had been infected. Then you had Dr. Nodder just doing what he does, just saying that there's genetic potential for hair sheep breeds in the US that have island descent in terms of disease resistance and to use them in breeding systems. So this is right around the time, the year 2000 is right around the time that Katahdin sheep are still listed as a rare breed in the United States, right? And now are the most registered sheep in the United States. So they've gone from being rare 25 years ago to now being that breed. And then Dr. Burke and Dr. Miller, Dr. Miller's down at LSU, parasitologist there, world-renowned parasitologist, that St. Croix sheep are more resistant than other hair breeds. So when I started at Virginia Tech, what Dr. Nodder, so Dave Nodder's a quantitative geneticist. That's what he does. And I'm fancying myself at the time as a quantitative geneticist. And he had a heart-to-heart with me in the hallway, and you know, maybe the math isn't for you, but the molecular biology fits you really well, right? So sometimes graduate students need that. They need a little bit of refocusing. He said, but I can't help you with that. That's not my area. So you're going to be on your own. So I took the challenge, went on my own and said, hey, we need to look at everything that's happening very early. And when I mean early, I mean days after infection. Within the first, so my involvement with St. Croix sheep started with my doctoral studies and then continued on in my work at West Virginia University. So here's what we found. This is what everybody found. Once infected, St. Croix sheep had lower egg shedding and fewer worms in their abomasum. And there was no discernible differences in their immune response at the time of egg shedding. Why? Because all the immune responses that mattered happened weeks earlier. So that's what we decided to do. We decided to go after those times that were earlier. So this is kind of the fundamental hypothesis and kind of the driving force behind the research that we do in our lab. That parasite resistance in St. Croix, as modeled by St. Croix sheep, is dependent upon an immune response. So once those animals generate an immune response, you get less larval establishment. So what St. Croix sheep do really well is they prevent larvae from establishing in the abomasum, which then prevents the establishment of adults. They don't mate, they don't have any eggs, pasture infectivity goes down, post-death goes down, everything else. Okay, so here's the validation of that. So my first study when I was just a new professor at West Virginia University, I made a student go out there and take St. Croix lambs that we got from Virginia Tech, because we didn't have our own flock of sheep. I called the guys at Virginia Tech and said, I want pregnant St. Croix ewes brought up to West Virginia. You got it, no problem. So we took those ewes, lambed them on an elevated floor barn, and we could guarantee that the naive sheep had never been exposed to Haemococcus at all in their life. Then the first time that we give them worms or a primary infection, look what happens in the red line. The fecal egg counts go up to 3,000 eggs per gram, and then they bottom out a week later. All by themselves, no dewormer treatment. I did end up deworming everybody at day 7 or at week 7, just because I'm a scientist and I'm going to do this. Then we come back and we give them a challenge infection. That's that green line across the bottom. Right? They're not out on pasture and they're not eating anything else. I can control everything, right? I'm a scientist. I'm about control, right? I need to take out all of these other extraneous variables to really get at the sense of what's happening. That's what I do. And that's what these sheep do. If you compare them to a susceptible breed, which we'll talk about, in our case, we're going to use the Suffolk as a susceptible breed. The reason that we use this in my research at WVU is that Suffolk sheep are accessible, cheap, and easy to find, right? Some of them, right? For sure, certainly. So our hypothesis here is that susceptible sheep don't have as good of an immune response. They allow larvae to establish. Adults are there. They're more fecund. And everything happens like that, right? Pasture infection goes up, GI and parasitism goes up, and it leads to the potential for host death. So if we look at these Suffolk crossbred lambs, don't worry about that drop. I'll tell you what happened there. Our naive lambs, the Suffolk lambs, were born on the same facility, all the same age. They get double. We didn't give them any more worms. We gave everybody the same amount of worms. We give them 10,000 larvae. That's our standard dose, is 10,000 larvae. If you look at their primary infection, they go up to about 6,200, something like that, and they stay pretty infected. This right here is when feeding goes bad. So if you've got a group of staff that don't want to put hay in the bunk and they just take a bale of hay and throw it in the pen with the sheep, what happens to everybody? They get diarrhea, right? All right, what happens to fecal egg counts when you have diarrhea? They go down, okay? They come right back up. So they never cleared it by themselves. We had to deworm them to get them cleared again, reinfected them, same thing, yeah.
So you put worms and infection in at the same time, or you infected them?
I give them worms right here.
Right.
1, 2, 3. Yeah. 21 days later, fecal egg counts go up.
Right.
Right.
And the green is the infection?
The green is the second infection.
And so you do that.
So I had 3 different groups. I had a group of naive lambs.
Okay.
I had a group of the first time lambs, okay, and I had a group of challenge lambs that received an infection. We cleared it, came back, give them another infection.
Okay.
That's what we did. Okay, pretty simple. Susceptible sheep can't clear the parasite by themselves. And what we're seeing before is what normally should happen. Your immune system should be able to do this. So what's happening in the sheep in this early infection that could be responsible for resistance? And here it is. So this is data from my dissertation project using St. Croix, and a 3-way cross is our wool breed, which is Rambouillet, Finn, and Dorset. So it's Dr. Notter's version of a polypay. He just left the targies out and did it himself. So his big claim to fame was reducing the length of estrus or anestrus period. So he took a sheep that had a 60 to 90 day anestrus period and dropped it down to 11 days on average. So you could breed sheep year round. We used those sheep as our susceptible model here and infected and looked at infection levels at day 3, 5, and 7 after infection. And this is after a challenge infection. At the time, we didn't, where we're at in Virginia, we didn't have the facility to have naive animals. So what you're looking at in those purple boxes up top is a cross section of the folds of the abomasum. So the abomasum has these little folds in there. We took one of those folds off every single lamb that we harvested that were either uninfected, day 3, 5, and 7. And that's what we see. So what you see in the first panel there on the upper left-hand side is a cross section of a normal animal that's not infected, right? You can see that in this area right here, I'm going to use this pointer instead of my finger. Ooh, slow down, mouse. This area right here will open up when cells are coming into that tissue. You see everything's nice and tight and pushed together like that. You can see right here in this is one of those wool sheep or the 3-way crosses at 7 days after infection. This area is opened up a little bit and you see all these dark purple cells that are right there, right? This is all taken at the same magnification all the way across. This is St. Croix sheep at 7 days after infection. So you're getting a massive influx of cells to the abomasum by 7 days after infection. So what I and an undergraduate did, so we had multiple slides for every animal. Every animal had 3 different slides. We took 40 non-sequential views off of every sample. This was about 49 or 50 lambs that we did on this study. So 50 times 3 is 150 slides, 40 times on each slide. You can kind of get the craziness that went on and develop counts for globule leukocytes, eosinophils, and neutrophils that were present in those tissues. So we're not seeing a lot of information with globule leukocytes. Eosinophils do what eosinophils do. They respond to allergens and worm. And we're not getting that much of a difference between the different breeds. But if you look at neutrophils, which we never expected to see there, or never kind of hypothesized that that was going to be the case, we kept seeing a lot of neutrophils in that site. So we almost get this semi-exponential increase in neutrophil accumulation at the site of infection by 7 days after infection, which Dr. Zajac would keep saying, Scott, you need to do more with this. Scott, you need to do more with this. So I remembered that lesson after I got back from my postdoc and ended up at West Virginia University and started looking at that. Now, the other thing that we can look at is lymph node weight, or the draining lymph node development after infection. Right there in the abomasum is kind of shaped like this, like a bean. On the lesser curvature of the abomasum, there's all of this fat and tissue that's right there. Inside that web of fat are a bunch of draining lymph nodes that treat that area, right? So that respond to different infections in that area. So we looked at the number of palpable lymph nodes that were there on every lamb when we killed them. We weighed their entire lymph node weight. So notice that there's no difference in the number of lymph nodes, but the weight, total weight of the lymph nodes that were there goes like this. It increases. Why does the doctor do this when you go in there and complain about your sore throat, right? They're palpating your lymph nodes. We went out, we had some reviewers that were being fussy with us. So we had lymph node sections. I had my microscope and we traced the outside area of those and figured out the lymph node area to calm everybody down. Same thing, right? So those lymph nodes are expanding in response to this, meaning that there's better antigen presentation that's going on there. So we're talking about neutrophils and their movement to this. And I've had a series of really bright and dedicated students come through the lab over the last 14 years I've been at WVU. And one time we were trying to think about if neutrophils are moving to the area, right? That's important. But it's also important what they're doing there. So we wanted to see if neutrophil movement was impacted by the antigen. So I remember I told you that Haemococcus produces different molecules that suppress immunity. Some of those molecules suppress migration. So we had the opportunity to test this $150,000 insert that goes into your incubator. And it's basically a mobile microscope that can scan all the wells on 6 different plates and do it as fast as you wanted to relative to the time period that you're going to do. So what we're able to do is at the bottom of these wells, we would put Haemococcus contortus crude larval antigen. This is basically, we took up Haemococcus contortus larvae, ground them up, took the soluble antigen, soluble fraction of that, and put it in these wells. Then on top of that, we just put an amount of neutrophils extracted from these sheep that had already been exposed to parasites. So what we found, anytime that this normalized cells per well gets lower, that means that the cells have left the well. They have migrated through a membrane that they have to physically push themselves through. They can't fall through it just by gravity. They have to migrate through that. This hair lambs, right? That's the HCLA, or the hair line, those are the St. Croix, are much lower than the suffix, right? But when we do nothing at all, the suffix move all over the place. They're moving up and down. It's really weird, right? It's like they have ADD. Then when you give them something that they should be paying attention to, oh, nope, I'm going to ignore it. And that's what happens. They stay up there and they ignore it. So we used that $150,000 machine. We took advantage of the free use of that for about a month, gave it back to the company, said thanks, but no thanks. We got enough data from this. We're off to the races.
And your wife progress.
Correct, right? So, the university didn't have to pay for it. So we made up our own migration assay. So you can buy these little inserts that go into wells, and then only cells that are big enough to fit through there, actively migrate through there, will move through there. So I had another bright kid that came from the school that I went to in Northern California, Chico State. It's one of the students I was able to recruit out to West Virginia. She did a whole study looking at kind of things that are driving neutrophil chemotaxis. So we know that in this crude and these larval antigen, or what the larvae produce, are neutrophil inhibitory factors. All coming back to neutrophils, right? So there is neutrophil inhibitory factor that is made to Ancylostoma caninum, which is the hookworm in dogs. We have this. I can go to the store and buy this, right? But I don't have this for Haemococcus. So I'm going to say that, okay, they're close enough, right? It's a neutrophil inhibitory factor. What that does is that also affects, you guys hearing me okay? Okay. It binds this receptor that's on the cell surface called CD11B. CD11B is integral in cellular migration through tissues. So what we did is we set up this experiment where we add HCLA at the bottom of the cell of the wells, put the cells on top, and look at percentage of cells that migrate. So the kids got to get in there and count, right? All the cells that are there. Crazy, right? That's what she did. Actually, we didn't count them. We labeled them with fluorescent dye and then did all this fancy stuff so we could not have to do that. So HCLA, when we put the NIF in there, look at what happens. When you put neutrophil inhibitory factor, it reduces migration, right? It reduces migration to the same level as CD11B. And if I put all 3 of them together, I don't get any kind of synergistic effect. It's the same thing. So we're pretty much convinced that that's what's going on. But HCLA is complicated because it's a mix of all sorts of different things that are in that worm. What we can do is extract what's called excretory secretory protein. So these larvae have what's called an excretory secretory pore that's just about, I don't know, a few millimeters down from their mouth. So if you take larvae and cause them to exsheath, so all these larvae, when they're out in the pasture, have sheaths around them, I can make them exsheath if I bubble CO2 into a solution that has that, put a cap on it, seal it, put it in a shaker in an incubator overnight, and they'll slip their sheath out. Then I take those larvae, bring them back into this well, and just let them sit there for a little bit, and they will express ES left and right. Then I can isolate that. Then I got ES. So we take a known amount of ES, we put that in there. Look at what happens with the suffix immediately. Just with ES alone inhibits migration. ES with the St. Croix are blind to it, right? But then when I add the ES plus the NIF, look at what happens to the St. Croix. They drop down and they stay there, right? All the way across. So what that's telling us is that the St. Croix are susceptible to the NIF, right? But with the ES alone, this reduction here is because of an NIF. And in this case, CD11B, and it's binding to CD11B, kind of cool, just different take on those type of things. All right, so if the neutrophils aren't migrating there, it doesn't matter what they can do. But if they are migrating there, and we are seeing other levels of different cytokines that are produced, then it changes everything. So I had a kid from New York City that, the whole thing, right? Comes out of Manhattan, first day on school. I always give my graduate students a hell day. Their first day at school is terrible. It is absolutely awful. His was shearing sheep with me. So he shows up in Toms and capri pants, right? And I said, you know what? I've got some boots in the back. You can use those. I had him packing wool all day while we were shearing some other sheep for other experiments. This kid went on, he's actually just applied for a job. He did a postdoc at UNC at the medical school there and is off doing some stuff for the Army now. What he was looking at is IL-4, or interleukin-4. It's a cytokine or a chemical that is produced by immune cells to initiate a certain response, particularly to parasites. That's what IL-4 is used for. If you look at these lines, we look at primed hair, so that's hair or sheep that have been exposed to a parasite before. When they respond to either CLA, which is crude larval antigen, or CWA, the only lines that come up are the ones for which ones, right? We have primed hair in the blue, and then the primed wool to CLA, but not to worm antigen. So both of the breeds can respond and produce IL-4, but if you're not there and you didn't make it to the site of infection, what's it matter what you can do, right? So that was the big thrust of his work, is kind of taking a look at that part of it as well. So we're able to use St. Croix sheep in that particular model as well. So if we look at gene expression, and I apologize for all of the acronyms on all of this stuff, we'll go through it. CXCL1 is a neutrophil recruitment gene, or a gene involved in that. MCP1, monocyte chemoattractant protein, brings in monocytes and neutrophils. IL-4 and IL-5 are common features of responses to parasites in a variety of different species. And what you can see here is very simple. It's non-detectable in the suffix sheet, and it goes up exponentially in the St. Croix, except for IL-5, it just bounces around a little bit, okay? So we have a bunch of IL-4 there. Well, we were really concerned about this IL-4 stuff and what's driving it and how it's working. And we didn't find the classical markers of upregulation of IL-4. And that would be, there's a signal transduction activating transcription 6 or STAT6 gene that's involved in all of this stuff. It wasn't showing up in any of our assays. And then we started going through the literature a little bit, and some other kind of large scale whole genomic analysis revealed this other protein called NOD-like receptor protein 3. And what NLRP3 does is it is involved in a lot of inflammatory reactions. But the title of the paper that we got attracted to was NLRP3 moonlights as an IL-4 activator, right? So we took a look at the transcript of NLRP3 and compared St. Croix and suffix and did this whole prediction model. And what you see is a difference in the predicted protein structure of these two molecules. Let's see if I just do this and I do this. And as they rotate, it's kind of cool stuff, right? You can see clear differences between the two predicted protein structures, right? That hook is a classic feature because what NLRP3 does is it develops a polymer and they polymerizes around these different areas, and that actually serves as the target that is identified and activates other transcription factors. So we wanted to evaluate NLRP3. So I was talking about the kid that's from Manhattan. He stayed around, did a PhD project working on this stuff. So how do you evaluate NLRP3 in this scenario? Everything's in vitro. So we keep a group of sheep that are there that are primed or at least have been exposed to parasites, and we just pull blood from them at will, right? So we go out there, if we need cells for something, boom, we snag them, bring them into the lab. If I just take complete media or Homunculus contortus larval antigen, you can see that there's breed differences in IL-4 production, right? I've already shown that to you ad nauseum. But then what we wanted to do is to see what happened to IL-4 production when we inhibited NLRP3. And come to find out, at the store, the science store, you can buy an inhibitor to NLRP3, pharmacological inhibitor, that I can put into the mix and do that. And in fact, that's what it does to the St. Croix sheep. But look at what it did to the suffix. Nothing, right? It did nothing to them, which was puzzling. So that made us think a little bit more. So we went in and did a few more studies with this. If you start looking at gene expression of STAT6, remember I was talking about that one before, look at what it does. When I give the suffix sheep NLRP3 inhibitor, they blow up. They were producing so much NLRP3 that it blocked everything. And the second that you suppress that, it allowed them to respond normally. So by studying parasite resistance in St. Croix sheep, we found out some of the mechanism of parasite susceptibility in Suffolk sheep, and that's what we see. So IL-4 goes up, IL-10 goes up, all of these things. So the inhibition of NLRP3 has a restorative effect, right? Or restores the ability to do protective immunity. Now, the follow-up study to this would be going out there and feeding sheep MCC 950. It's too expensive, right? It's just crazy. We did this, that's enough. Okay, so what does all of this mean when we put this together? And I'm sorry, I can't talk very long, right? Because I didn't want everybody to fall asleep, and I knew that Dr. Willius would have an abundance of slides, so I wanted to keep this short. What does this mean? What does all of this immune response result in? This is the number of larvae that are in the stomach of sheep 7 days after infection. 16 on average and 530. Do we need to do statistics on that? No, right? We're done. We're done, right? That's it. Now, everybody had either gone way past what we did in terms of the time of infection or real early like us, right? We weren't the first people at the dance on this, but we were the ones that did the most with it. But there was this gap in between everything. It was 10 days. So I had another student that was one of my first kind of cohorts of graduate students. She's a trip. But her whole thing was doing this work. So we killed lambs at 10 days after infection. What we found at 10 days after infection is that finally the wool sheep started experiencing lymph node hypertrophy. So you can see a difference right there. It's peaking right above the line there of wool and hair sheep. And actually, if you look at this value compared to, where's my pointer? There it is. I gotta go slow. This value compared to the hair sheep, it actually comes out at about day 5 of the hair sheep. The hair sheep are the same as the wool sheep in this case because their lymph nodes are getting smaller. They're done. We're done with this. We're moving on, right? They're done with this. And then we look at IL-4 because The uninfected animals didn't have anything. The challenge ones didn't have anything. Neither did these ones. The only thing I can do is ratio it. Finally, 10 days later, am I getting IL-4 production from the Suffolk sheep? Took us a while to get there. So what have we observed in all of this stuff? We had a big year that year. We're just publishing like crazy. Reduced abomasal worm burden by day 7 in St. Croix sheep. Lymph node hypertrophy by day 7 in St. Croix sheep. Abomasal IL-4 expression is upregulated between day 3 and 5. And we've got an abomasal influx of neutrophils between day 3 and 7 in St. Croix sheep. So effectively, we can say that there is a 7-day delay in the onset of IL-4 production in Suffolk sheep, and there's a 5-day delay in all this. What happens with delays in these sheep that are constantly consuming larvae? It spirals, right? Okay, it's simple, right? Now that we've done all of this stuff and we've done all the labor on all of that stuff, it's really simple and really straightforward. So St. Croix sheep are just more immune competent, right? So how do we test that? I've showed you with parasites. Great. That's cute. What else? All right. So how do I model a bacterial infection in livestock without initiating a BSL-2, BSL-3 nightmare of doing all of that stuff in my lab, infecting animals, controlling where it's complicated. I can replicate this by just using something that is on gram-negative bacteria called lipopolysaccharide. If I took lipopolysaccharide and throw it in Yates' face right now, about 30 minutes later, she's going to get real sick. And about an hour later, she's going to have a fever that's going to go up 2 to 3 degrees. That's simple, right? And that's going to be mediated by all of these immune events that are going on in their body. So instead of throwing it in their face, I just give it to them in their neck based on how big they are. And that's what we did. So we set up this massive experiment at the farm where we've got, now we added some Katahdins in here, but I'm not going to talk about those. We had St. Croix, we had 10 St. Croix, 10 Suffolks, and then 10 low EBV Katahdins and 10 high EBV Katahdins. We did serum analysis, blood and temperature and videos at all of these different time points all day long, right? We didn't mess with the sheep in between there. We took all this video to kind of get a sense of what the animals look like, how they were behaving with this. So how do you score animal behavior? Well, somebody came up with this really cute sheep grimace score. So they look at something called orbital tightening, which is this, right? Squinting. Their head and neck and ear position. So my dad would always say that a sheep's ear are a window to their soul, right? And you can tell what's going on with them. If their ears are up, right, they're okay. If they're down, there's a problem. Head and neck position, when they're ewe-necked and their head's down, that's a problem. And then Fleming response. So you can use this score to do this, right? So we had videos at the bottom end of every hour for 12 hours. We took a kid, trained this kid on how to do grimace scoring, tested all of that. He's good to go. All right, sit here, Bubba, and watch all of these videos and score every group of these animals. I use a lot of undergraduate labor, right? I use them, and I put an emphasis on use because they don't get anything from it. They're just there. That's what we do. Made the kid do all that stuff. This is what we found, which is really cool. 2.5 hours before, everybody's normal. We're fine. Everybody's okay. 30 minutes after we gave them the LPS, the Suffolk sheep already start to look bad, right? So their grimace score goes up to an average of 2. Then it gets worse because what's going on with those sheep? They're getting a fever. What happens when you get in a fever? Are you super spunky and lethargic? Or super spunky? That's an oxymoron. Here we go. Are you spunky and moving around? No, right? Down, don't feel good. That's what those Suffolk sheep look like. I forgot to put the videos in here, but the Suffolk sheep look like absolute hell. Yeah, I did at the other one. I forgot to put it in here. They're terrible. They're down like this. They're moaning and grunting, and their backs are arched. We actually had a couple of the Suffolk sheep die. Cytokine storm, right? That came up through all of that stuff that happened. But what you can see with that grimace score is that about 5.5 hours later, we're okay. Everything's okay. The Croix were eating feed again. Everything was okay. If you look at their temperature curve, which I didn't put in here because it's not super sexy, it stays pretty flat. They really don't respond to much. The Suffolks get a temperature, right? But the Croix just stay flat. They get sick, they get over it, they're okay. What drives treatment of animals for illnesses? Symptoms, right? That window to their soul, how they're behaving. The longer they're showing these symptoms, the more likely they are to receive a treatment for some disease. Okay, so let's go into some of the stuff that's going on with this. So we looked at white blood cell count. Well, all the animals, when they get LPS, become lymphopenic. Basically, all the white blood cells in their body get shunted to their liver, right? They just fly there. And you can see how fast that happens. That happens between when they received it to 1 hour after they got the LPS. Everything just flies there. And then slowly, those cells, it's not like we're making new cells that are coming into tissue. Those cells are coming out of the liver and going back into circulation. So they're recovering. But look at how fast these ones are recovering compared. There's a delay, right? There's a constant theme here, regardless of the pathogen that we're talking about. So we're getting a difference in white blood cell count that's largely driven by what's happening with neutrophils in that particular case. All right. If we look at tumor necrosis factor alpha, yeah, this is busy and it's crazy and it's a little out of control. Just look at up to hour 3. Everything's going okay. Then the Suffolks spike. So do the St. Croix. They get a little sick, then it drops off. TNF alpha is what drives some of the HPA axis or the hypothalamic pituitary adrenal axis to start causing body temperatures to go up because bacteria don't like high temperatures. Okay, well, there's a crossover point. And you see that in the middle, there's an X. Now, scientists get really excited when there's X's in data like this, right? That means that there's an interaction that's occurring. One of them's going down, the other one's going the other direction. Okay, those stupid sheep out here, these Suffolks out here are still have higher levels of TNF alpha in their blood. They haven't cleared the response then. The Croix are back down to about where they were when they started. These sheep aren't in some fancy facility. They're outside in a barn, right?
Are you
Okay.
there?
So then we finish with this one. So that we also work with a stress physiologist at the med school. And he said, hey, did you got serum on all those sheep, huh? I said, yeah. He's like, put it down on a plate and see if there's any hemolysis in it. Like pool them all together and see what happens. Okay, we did. Wow, right? Okay, so typically when haptoglobin levels go down, that means that there's some hemolytic event that haptoglobin is responding to. It's binding hemoglobin because blood cells are popping.
So right now,
that's what happens in the Suffolk. I don't know why they're excessively high there, but it continues to go down. The Croix are flat line all the way through there. But what you can really see is this is, my student does a good job with these pictures.
So he's doing a study between Suffolk and St. Croix.
Going through here,
but I don't really
understand the darkening of the wells as you go across the plate.
Oh, and the whole
These are
thing's
the
recorded
Suffolks,
on
these
the
are the
St. Croix website.
Then what we did to quantify that is we just put it on a plate reader, measured optical density of the wells.
What's going on right now?
See how high it goes up here, all the way across.
Okay, I'm listening and trying to do my stuff at the same time.
Okay. All right.
Oh, no. I don't hear anything. Okay.
So can we say then that the St. Croix breed has immunological superiority, right?
Okay.
This is my message for the rest of this.
Oh, okay.
Is that this trait must not be lost in
or
this breed.
fish or whatever.
You guys are the caretakers of this trait because there is no other breed that responds like this. Dr. Wildius did all of this work. You see all the work that all of these people have done across their careers to demonstrate this.
We could have
this trait must not be lost in search for something else.
Let me see if I think there's 2 years
because we've seen it before. All of these sheep started out having some sort of immune competence,
one of those candy bars.
right? We have effectively bred it out
I
of
had
most of the
raisin
sheep in the
toast for
United
breakfast
States,
because the other toast is all gone.
except for St. Croix.
And I ate the rest
Right.
of the ricotta cheese.
Careful balance should
okay.
be considered
So
when
now I can
selecting
start
for
working
other
on, I got
traits.
some
See
yogurt.
Suffolk sheep.
Okay.
The selection and emphasis
Okay.
on growth and carcass
So
merit
we have-
has
okay, I'm
blind,
gonna-
and
I'm gonna-
then
there's some
couple
carrots
that
and
with
we're chopping
really
it up.
effective
I don't like
antimicrobials
those carrots.
and really effective
And
anthelmintics.
that dump that other
We're
carrot.
blind
stuff in.
to immune response.
But-
We're blind to an animal's ability to survive in that because we
Okay, I'm
have
gonna do that tonight. I'll
taken
do it
care
tomorrow.
of them.
Okay, all righty.
This is something that we have to
I
do,
have no
right?
idea. don't-
I think that
I've been
for
doing
St. Croix
all the-
breeders, you have an extraordinary opportunity
I gotta
at your
go.
fingertips
all righty.
for your sheep to be used in
I'll be
crossbreeding
careful,
systems
okay?
because they bring
Oh,
a
you
unique
work
set
tomorrow?
of traits that
Okay.
cannot be found in any other breed. I don't care how parasite resistant a Katahdin is, it is still not as parasite resistant as the average St. Croix. That's why during the upgrading process of St. Croix, my advisor at Virginia Tech used a Suffolk and a St. Croix to upgrade our Katahdins, right? Used them both. But for this to be effective, you have to have maintenance of seed stock producers. You guys in this room are vital to this because you're the ones who are here. You're the ones who are producing the seed stock. How do you maintain the selection and the maintenance of that particular trait within your sheep? So have to do all of this stuff, right? Virginia Tech, if it wasn't for them, I wouldn't have any sheep to work with. I wouldn't have any parasites. Dr. Zajac gave me my first dose of parasites. So I still have some of her worms that I give to other people and that's polite. I sell to other people, right? Because I'm an academic entrepreneur. The Northeast SARE, NCVP, USDA, the American Sheep Industry, just various funding organizations that have helped us out along the way. And I mean us because it's all of those people. So, okay, questions. Let's have some fun with this stuff because we should be talking about things. Yeah, nothing. Yates, please.
Yes. Okay, so as a seed stock producer, how can I know that I'm maintaining, how can I know that I can maintaining this effect in my sheep?
That's a fantastic question that I am not trained to answer. So I am going to, yeah, I don't know. I'm going to obfuscate just like most academics do and say that, you know, there are other geneticists and other people that are better inclined to answer that question. I have some thoughts that I'll share offline with you, but anyway,
Okay.
there you go.
Any studies that have been done to see whether maybe chronic deworming has caused this in other species that it has been maintained in the St. Croix?
So you're thinking about the overuse of anthelmintics. There's no real study to do that. All we can say is that, hey, they don't work anymore. It's probably due.
So nobody's taken St. Croix and chronically dewormed them to see if they lose that? I mean, anecdotally, I've always heard.
Well, I don't think that they're going to lose. So you're not going to lose immunity by deworming the sheep. What you're going to create are a set of worms that are now resistant to the drug. Now, the great thing about St. Croix is because they have a myriad of immune mechanisms, and what I gave you was just a taste because I didn't think that you could handle the full Monty of this, right? Is just to focus on this one little wedge of this that seems to make a nice story, right? It's not just neutrophils. It's not just IL-4. It's not just antibody. It's all of it. It is a kitchen sink, robust, massive response that these sheep are generating that parasite isn't going to evolve resistance to. It's not a single point. The thing about the evolution of resistance in the worm can happen in 4 or 5 years. But with the immune response, we haven't seen that. Otherwise, all the sheep in St. Croix would be dead. It's an island population, right? No migration, little just getting mutation because of the inbreeding that's going on. All right. So do the sheep lose their ability to fight the worm because of deworming? Probably not. The worm is losing its ability, but their immune system's still going to be tested by a lot of different things. The thing that we're doing now to measure kind of generalized immunity is looking at antibody production to a booster vaccination of clostridial diseases. right? So your CD&T vaccine, 1 week after a CD&T vaccination, that's what we're measuring antibody responses on, because with the Katahdin sheep, we found that sheep that are very parasite resistant within the Katahdin breed have a higher immune response to the vaccine. So we're trying to see if I can take that information, get enough samples from everybody across the country, and back to your answer, I was supposed to answer offline, but here it goes, is to develop a breeding value for antibody production. That
Awesome.
would be the idea with the Croix, is look at that. So the more antibody, but again, it might be one of those things where you don't get a lot of variation.
Mhm.
Everybody's just high,
Just- okay.
right? Just like with
But it's
fecal egg
high
count.
on the people's
Right.
blood.
That's right.
Got it.
That's right.
Questions from our listeners?
Fantastic.
One person asked, when you gave the 10,000 strong giles orally, were those all verified barber pole, or were they, could there have been other strong giles?
So when I count, and so when I grow larvae that are homunculus. Do I get some other strongyloides in there, which are not strongyles, they're strongyloides. There are some strongyloides, but I don't count those, right? Does that make sense? So when I'm doing my dilutions on all that, I may get some strongyloides and I can't do anything about it. So more often than not, our set of larvae are very good at causing disease, right? And the story behind my larvae goes something like this. I got larvae from Dr. Zajac. Everything was going fine until a graduate student stopped maintaining the larval culture, kind of took a break from it. And I went to him, I said, and his name is Rush. I said, Rush, now it's Dr. Holt. He got it. He went to vet school, did a different sort of science. Rush, where are the larvae? He's like, oh, I kind of forgot to do them. We had no larvae to infect anything with, right? I had just purchased an $1,800 Suffolk ram. This thing had a rock star ribeye area. We were going to do some stuff with Suffolk sheep out there at the farm. Fantastic, great. The ram was dead 3 days later after I dropped him in the field with ewes to breed them.
Oh, no.
When I mean dead, right? He was laying up against the fence, dead at the bottom of the pasture that we had for those ewes. So the veterinarian that works in the school with me, she was going to go out, and I said, let's do a necropsy on this ram. Get all the students out. We open up this ram. He's still bleeding. This is about 6-7 hours, maybe even as much as 8 hours after the ram had died, right? He was bleeding. You cut him open, he was bleeding. Does that sound normal to you guys? He didn't have any rigor. You can move his legs around like this, right? So we both kind of looked at each other, puzzled. Everything on this ram was pale. Everything, right? Subcutaneous, he was pale. Heart, liver, lungs, kidney, everything pale.
Excellent.
We're looking at each other and like, I said, there's no way, Margie, there's no, Dr. Minch, Margie. So I said, there's no way this is worms. I know what I'm doing with worms. And she said, let's just take a look at his gut just in case. That's where it was. Shag carpet inside his abomasum with worms. And it is what's called, yes, it's acute hemoncosis. So Rush's penance for losing that was taking that stomach out of the sheep and then picking out all the female worms and gently crushing them up to release their eggs, growing those eggs, and then we take those larvae, drop them into some culture weathers that we keep on the farm, and that's where our larval culture started.
Oh, wow.
So he wasn't dead, he was just.
No, he was dead.
Oh, no. Well, he got bled from the inside out from something called acute hemoncosis, which I'd never heard. I'd heard about. It was in all of the textbooks. I'd never seen it. I had to pay $1,800 to see it. Okay, tax dollars hard at work with lots of mistakes from overconfident faculty. Other questions? Yeah.
So if they like parachute, take Croix and crossbow, how much of that resistance transfer?
That's a fantastic question. So with Katahdin sheep now, what we consider the heritability, right? So that's what our measurement of that would be, is about 25% of the heritability or of that trait is heritable, but it's going to vary. And it varies by environment. It varies by exposure. I think that level's probably higher in St. Croix sheep than it is in other breeds. So what you would see, and typically what other people have seen, is that when you take a Croix and you cross it with a Dorset, you get something intermediate. And when that happens, it's typically a highly heritable trait. But in that breed, that doesn't mean that it's the same way in a Katahdin sheep. So how much of that? Less than half is going to transfer over. And Mendel's going to screw you a little bit on that, right? However, he, independently assorts and segregates all your genes, you're going to get variation with that.
This was never really 50-50.
Yeah. What else? Yeah.
So I am in NSIP and I do fecal tests. Is there a target time? Like I should target wet seasons to collect samples to see if there's more infection?
I would target the time of year when your lambs are most likely to have a heavy infection.
I just did mine and it was towards the end of July and we were 3 weeks into drought.
Yep.
So if I'd have done it in June when I was in.
Or if you've done it in September, October, depends on where you're at.
Yeah, but so.
You're Pennsylvania. I wouldn't be afraid of sending one in September, October.
Yeah, so I could do it again.
Yeah, yeah. So, number one, fecal egg counts take some time to do, right? But you don't have to do everybody. You can kind of get a sense where everybody's at. Just even on FAMACHA scores, kind of get a sense if you're starting to see some variation in FAMACHA scores. That should give you a sense of what's going on and do that. The other thing particularly with post-weaning fecal egg counts, is that they're not tied to a particular time. All they are is post-weaning.
Because most of the results were 0, the highest was 500.
Right. So if you're a quantitative geneticist and you get data in from these people, what does a 0 fecal egg count mean?
They were shedding nothing today.
Are you sure about that?
There was.
Do you understand? Do you see why this is a problem? Maybe, or they're really resistant, or they weren't exposed to anything. You have no idea what a 0 means. That's why they put an average of 500 for the flock or the contemporary group that's being submitted, so that they can assume that there is a level of infection that everybody was exposed to. But you have sheep that are designed not to have parasites, right? So I think that might not be the best metric in the world for you.
So the better metric would be the thing we're not talking about.
Potentially, right? You could actually run into the same problem,
Fair enough.
right? But so I work with some breeders that are in Minnesota. They will take their fecal samples in October, September. Right. But if I got a fecal sample coming from Minnesota that comes in May, what have they been exposed to? They got nothing. So they're all 0. Like, oh, my sheep are all 0. I said, yeah, man, you sent me fecal samples in May. Of course they're 0. You weren't exposed to anything, right? The ground had just thawed out. So calm down, wait a few months. Let's do this again. The other thing is that there's a lot of genetic correlation set up within the system between post-weaning fecal egg count and weaning fecal egg count. So you're going to get a value for either one of those. You just take the sample when the animals are going to be most infected. That's the most important thing. And it's okay to not have a weaning fecal egg count. It's okay to not have a post-weaning fecal egg count. Get the one that lines up when the animals are going to be the most infected or the most likely to be infected. Fair enough. And if you need some worms, call me, right?
Yeah, Rush took care of that for us.
Well, yeah, he did eventually. That's for sure. That's for sure. Anything else? Well, thank you so much. It's been a pleasure talking with everybody. I appreciate your time and questions. I'll be around the rest of the afternoon.
Thank you.
Yeah.
So let's go ahead and take like a 5-10 minute break, set up the next